Imagine a castle under siege by an invading army.
Okay.
You've got the high walls, the heavily armed guards on the parapets. I mean, the entire defensive setup is fully operational.
Right. The defenses are up.
Exactly. But the invaders, they don't attack the walls, they don't use catapults or anything like that.
So what do?
Well, instead, they send out these tiny, invisible messengers that slip right past the defenses, sidle up to your castle guards, and just start whispering in their ears.
Oh, wow.
Yeah, and whatever they're whispering, it is incredibly effective. The guards are completely brainwashed.
They switch sides.
They literally turn around, put their backs to the invaders, and start actively defending the attacking army against any reinforcements you try to send in.
That sounds like a scene from, like, a fantasy novel.
It really does. But
Yeah.
this isn't fiction. This is the exact microscopic strategy utilized by an incredibly aggressive type of cancer to completely hijack your immune system.
It is a terrifyingly elegant biological strategy, honestly.
It really is. And, you know, our mission for today's deep dive is to unpack exactly how this happens.
Right, because we finally have the map. We're looking at a really groundbreaking study from the journal Cancer Cell.
Yeah, let's talk about the sources. They use some pretty intense technology for this, right?
Oh, absolutely. The researchers utilized high-dimensional single-cell proteomics.
Which sounds incredibly complicated.
It is, but basically it means analyzing 50 different tumor microenvironment proteins simultaneously. And they did this across both human tumor specimens and mouse models to just completely decode this brainwashing mechanism.
And more importantly, to figure out how to, you know, shatter that defensive shield.
Exactly.
Okay, let's unpack this from the very beginning. For anyone listening who follows oncology, you probably already know the stakes with TNBC, or triple-negative breast cancer.
Yeah, it's notoriously difficult.
Right, because it lacks the 3 standard receptors: estrogen, progesterone, and HER2.
Which means our standard targeted therapies just kind of bounce right off it.
Exactly. It's known for driving tumor heterogeneity and metastasis.
Yeah.
But what struck me right away about this specific study is the methodology.
The fresh specimens.
Yes. They didn't just look at a few cell lines in a petri dish. They used single-cell proteomics on 161 fresh, treatment-naive TNBC specimens.
And that phrase, treatment-naive, is absolutely critical here.
Right. It means these tumors hadn't been exposed to chemotherapy or radiation yet.
Because once you introduce chemo, you basically drop a bomb on the tumor microenvironment.
Right. The baseline cellular relationships get completely scrambled.
Exactly. So by looking at fresh, unbombarded tissue, the researchers could observe the natural, undisturbed ecosystem of the tumor.
Just seeing how it behaves in the wild, basically?
Yeah, and the technology, the high-dimensional single-cell proteomics, it's fascinating. It essentially allows you to tag 50 different proteins with heavy metal isotopes.
Oh.
And then you run millions of cells through a mass cytometer, mapping exactly which cells are expressing which proteins all at the exact same time.
So it gives you this high-resolution spatial map of the entire battlefield.
Precisely. And when they looked at that map, they zeroed in on a specific culprit driving the evasion.
The cancer stem cells, right? The CSCs.
Right. Now, we're all pretty familiar with the concept of cancer stem cells driving therapy resistance.
Yeah, usually we think of them as these dormant cells just waiting out the chemotherapy.
Right, but the way they operate in the specific TNBC model, it feels different. They aren't just hiding.
They're proactive.
Highly proactive. The old analogy used to be that cancer stem cells are like the queen bees of a tumor hive.
Okay.
But based on this spatial mapping, a better way to visualize them is as an invasive weed's deep root system.
Oh, interesting.
Yeah, they don't just sit there. They actively alter the chemical composition of the soil around them, you know, to ensure nothing else can survive except what they want.
Which brings us to the guards they're brainwashing. The proteomics map revealed this glaring pattern. Right. In the tumors that were highly enriched with these stem cells, what the study terms CSCs high, the local environment was absolutely swarming with regulatory T cells.
Specifically, the CD103 positive, FOXP3 positive subset.
Right. And for those listening, Tregs are normally the immune system's peacekeepers.
Right. Exactly. They normally step in after an infection is cleared to suppress the CD8 positive killer T cells. They act as a brake so your immune system doesn't accidentally trigger an autoimmune disease.
But the mapping showed the cancer stem cells are actively recruiting these peacekeepers to build a localized safe zone.
Yes. the researchers proved that right in the immediate physical proximity of these cancer stem cells, the Treg concentration was massively elevated.
And CD8 killer cells.
Completely absent.
Wow. So the cancer is purposely breeding peacekeepers in its local neighborhood to stop the immune system's riot police from doing their job.
That's exactly what's happening, and that spatial correlation is what drives the accelerated progression of TNBC.
It creates this impenetrable immune-suppressive bubble.
Right. But, recognizing that the bubble exists is really only step one. The real mystery here is the communication method.
Yeah, how does it actually do it?
Well, think about it. Put yourself in the position of that cancer stem cell for a second. You're anchored in a dense extracellular matrix. You need to brainwash a naive T cell that's floating nearby.
Right.
How do you actually get incredibly complex biochemical instructions across that physical space?
Okay, before we answer that, let's do a quick recap for the listeners, just to lock in these concepts. We want to make sure every detail is clear.
Great idea.
So, bullet point number 1. TNBC is a highly stubborn cancer that lacks the standard targets, making it super aggressive.
Right.
Number 2. The villains here are the CSCs, the cancer stem cells. Think of them as the deep toxic roots of a weed.
Yep.
And number 3, the Tregs are the immune suppressors, the brainwashed castle guards that the tumor recruits to protect itself from the killer CD8 cells.
Perfect summary.
So here's a question for you listening right now. If you were that anchored cancer stem cell, how would you physically send a signal across the cellular space to build this protective shield?
It's a great question.
And the answer is, you send out biological drones.
Extracellular vesicles, or EVs.
Right. We know tumors use EVs to communicate. And a good visual mnemonic for you listening to remember EVs is to think of them as biological envelopes and vehicles.
Oh, I like that. Envelopes and vehicles, because they literally package things. Tumors pack these nanoscale lipid bubbles. We're talking 40 to 200 nanometers with mutated RNA and proteins, and deploy them into the microenvironment.
But usually with standard protocol, what we call canonical signaling, the recipient cell has to physically swallow that EV, right?
Exactly. Through a process called endocytosis. It brings the package inside, the bubble pops, the RNA is released, and the cell is reprogrammed from the inside out.
That's the canonical pathway.
Right. But what's fascinating here is the massive plot twist the researchers uncovered.
The non-canonical surprise.
Yes. The EVs deployed by these specific TNBC stem cells, they do not rely on internal cargo to reprogram the T cells.
Wait, really?
They don't need to be swallowed at all. They bypass the endocytic pathway completely.
Okay, let me stop you there, because biologically that sounds incredibly difficult to pull off.
It does.
You're telling me a lipid bubble just drifts over, bumps into the outer membrane of a T cell, doesn't transfer a single strand of RNA inside, and somehow manages to completely rewrite the T cell's functional DNA to turn it into a Treg.
That is exactly what happens.
How does a purely external physical touch reprogram a cell's nucleus?
It's all about membrane topology and non-canonical juxtacrine signaling.
Juxtacrine signaling.
Yeah. The entire brainwashing sequence is triggered by a specific integral membrane protein embedded on the outer surface of the EV.
And that protein is called TSPAN8.
Correct.
Okay, so if we go back to our analogies, it's not a Trojan horse where the soldiers have to get inside the city walls.
No, not at all.
It acts like a skeleton key.
A floating skeleton key, precisely.
Just bumping into locks.
Right, and the researchers proved this definitively. They engineered synthetic liposomes.
Which are what, exactly?
Basically, completely empty lipid bilayers. They had absolutely zero internal tumor cargo inside them, just empty bubbles.
Okay.
But they embedded just the TSPAN8 protein on the outer surface of these empty bubbles and introduced them to naive T cells.
Let me guess. The empty bubbles still brainwashed the T cells.
Instantly.
Wow.
The physical engagement of TSPAN8 with the outside of the T cell was sufficient to induce the full immunosuppressive Treg phenotype.
That is wild.
It is a terrifyingly efficient mode of communication because it skips the energy-intensive process of endocytosis entirely.
It's the biological equivalent of, like, walking past a building, scanning an RFID badge on the outer security panel, and instantly locking down the entire facility without ever opening a door.
That is a perfect way to visualize it.
Okay, so let's do another quick recap before we dive into the microscopic circuitry of that lockdown.
Sounds good.
Bullet point 1. EVs, our envelopes and vehicles, are tiny biological drones used for cellular communication.
Yes.
Number 2, canonical signaling involves swallowing the drone to get its internal cargo.
Right.
But number 3, Non-canonical signaling in this breast cancer relies purely on a surface protein called TSPan8, which acts as an external key. No swallowing required.
Spot on.
So, for the listener, think about your own daily communication. Is it faster to send a long, detailed package that has to be opened and read, or to simply walk over and press an emergency button on a control panel?
The button is always faster.
Exactly. And that brings us to the lock, the emergency button itself. TSPan8 is the key, but what is it actually unlocking?
TSPan8 actively seeks out and binds with incredibly high affinity to a receptor called CD103.
Which is on the surface of the naive T cells.
Yes, also known as integrin alpha E beta 7.
Now, CD103 is an integrin, and usually an integrin's primary job is cell adhesion, right? Like helping cells stick to things.
Exactly. But in this context, it acts as the primary signal transducer.
Okay, here's where it gets really interesting. I want to trace the intracellular dominoes here. We hear terms like signaling cascade or kinase recruitment in these studies, but let's explain the actual physics of it. No detail left behind.
I love it. Let's get into the mechanics.
The key, TSPan8, slides into the lock, CD103. What is the immediate physical consequence of that connection?
Okay, so when TSPan8 binds to CD103, it causes a conformational change in the receptor.
It literally changes shape.
Yes. Now, CD103 itself lacks kinase activity. It doesn't have the chemical machinery to ignite a reaction on its own inside the cell.
Okay.
Instead, that physical shift in its shape exposes a binding domain on the intracellular side of the membrane, and this acts like a biological magnet.
Ooh, a magnet. What does it pull in?
It aggressively recruits a specific protein kinase from the cytoplasm called MAP4K4.
So MAP4K4 gets pulled up to the inside of the cell membrane, and kinases, their whole job is to phosphorylate things. They tag other proteins.
Exactly. Phosphorylation is how cells physically flip their internal switches.
Let's break that down, because adding a phosphate group isn't just like putting a sticker on a protein.
No, not at all.
MAP4K4 takes a negatively charged, bulky phosphate group from an ATP molecule and forces it onto a specific target.
And that negative charge violently repels other negative charges within the target protein's own amino acid structure.
Right. It physically forces the entire 3D shape of the protein to contort and bend.
It's like pulling the pin on a grenade. The contortion exposes the active functional site of the protein.
Okay, so what is MAP4K4's specific target here?
The target is a well-known tumor suppressor protein called LKB1.
But it doesn't just hit LKB1 anywhere. Location on the protein structure is everything, right?
It is highly specific. MAP4K4 phosphorylates LKB1 precisely at the serine 428 residue.
Serine 428.
Right. And to do this stably, it utilizes a heterotrimeric complex involving two other proteins, steroid and MO25.
So it's a team effort. And when LKB1 is phosphorylated at serine 428, it morphs into its active state.
Yes. And once active, this hijacked tumor suppressor knocks over the heaviest domino in the entire cellular pathway. It activates AMPK.
AMPK, that's the cellular energy sensor, right? The metabolic master switch.
Precisely. AMPK regulates lipid and glucose metabolism.
But in the context of a naive T cell, flipping the AMPK switch initiates a massive metabolic reprogramming.
Exactly. And that reprogramming culminates in the massive upregulation and stabilization of FOXP3.
FOXP3. And there it is. The holy grail of regulatory T cells.
The master switch.
Right. FOXP3 is the master transcriptional regulator. Once FOXP3 is elevated, the cell's fate is sealed.
It is officially functionally transformed from a standard T cell into an immunosuppressive peacekeeper.
Working exclusively for the tumor. The brainwashing is complete.
The initial phase is complete, but the study highlights a deeply insidious twist to this mechanism. The signaling cascade doesn't just hit FOXP3 and stop. No. It creates a vicious, self-sustaining loop.
Oh, right. Because FOXP3 doesn't just sit in the cytoplasm. It goes straight in the nucleus and binds to the cell's DNA.
It transactivates the gene ITGAE.
ITGAE.
Yes. And do you know what the ITGAE gene codes for?
It codes for the CD103 receptor, the very lock that started this whole process.
Exactly.
Wait, so it's a biological echo chamber. The cell gets the signal to become a peacekeeper, and its very first act is to build more antennas, more CD103 locks, to receive more of that exact same brainwashing signal.
It's a massive positive feedback loop. TSPan8 hits CD103, MAP4K4 activates LKB1, which triggers AMPK, which produces FOXP3.
And the FOXP3 builds more CD103. That is wild.
And this loop is exactly what drives the massive, rapid clonal expansion of Tregs in the tumor microenvironment. It explains why the proteomics map showed the cancer stem cells completely saturated by these peacekeepers.
Unbelievable. Okay, it is time for recap number 3. We just covered a lot of molecular ground.
We really did.
So bullet point 1, CD103 is the lock on the T cell.
Yep.
Number 2, MAP4K4, LKB1, and AMPK are the internal dominoes that get knocked over when the key turns the lock.
Right, the internal tripwires.
And number 3, FOXP3 transforms the cell into a Treg and forces it to build more CD103, creating a brutal positive feedback loop.
A perfect summary.
So, to you listening right now, looking at this vicious cycle from TSPan8 on the outside all the way down to FOXP3 on the inside, where would you try to break the chain to stop the cancer?
That's the million-dollar question for oncologists.
Right, because tracing a map and winning a war are two different things. How are researchers utilizing this incredibly dense mechanistic insight to actually help a patient sitting in an oncology ward today?
Well, the most elegant interventions are often the most upstream.
Meaning you target the very beginning of the process.
Exactly. If you can stop the key from turning, the entire cascade collapses. So the researchers developed and utilized preclinical trials with an anti-TSPan8 monoclonal antibody.
Neutralizing antibody, so it targets the EV itself.
Right. This antibody is designed to bind specifically to the TSPan8 protein on the surface of the tumor's EVs.
So it basically coats the floating keys in biological superglue.
That's a great way to put it. It completely neutralizes them so they can no longer dock with the CD103 locks on the naive T cells.
Wow. And if the key is blocked, the CD103 doesn't shift, MAP4K4 is never recruited, and FOXP3 is never produced, the echo chamber just goes completely silent?
Exactly. But there's a catch.
There's always a catch with TNBC.
Right. With treating aggressive cancers, a single therapeutic wedge is rarely enough. You might stop the brainwashing, but you still have a massive, aggressive tumor sitting there.
Right. You need the riot police to actually go in and clear it out.
Which is exactly why the true clinical breakthrough in this study is based on combination therapy.
Synergy.
Yes. The researchers combined the anti-TSPan8 antibody with an existing class of drugs known as immune checkpoint inhibitors, specifically anti-PD-1 therapy.
Now, PD-1 inhibitors are kind of the rock stars of modern immunotherapy. But as we know, TNBC is notoriously resistant to them.
Highly resistant.
And it makes sense why, right? Because PD-1 inhibitors rely on CD8 killer T cells actually being present in the tumor to do the killing.
Exactly.
If the cancer stem cells have built a massive Treg shield that physically excludes the killer cells from the neighborhood, the immunotherapy drug has absolutely nothing to work with.
Right. It's like handing high-powered rifles to soldiers who aren't even allowed on the battlefield.
Great analogy. So how did they test this combination?
To test this, the researchers used a highly specific in vivo methodology, the 4T1 syngeneic mouse model.
Okay, let's break down why they chose that specific model.
Yeah.
Syngeneic means the tumor and the mouse share the exact same genetic background, right?
Right. Unlike some models where human tumors are grafted onto immune-deficient mice, a syngeneic model features a mouse with a fully intact, fully functioning immune system.
Which is absolutely critical when you're testing an immunotherapy drug. You need a functioning immune system to see if you can manipulate it.
Exactly. And the 4T1 model is famously immune cold.
Meaning it naturally excludes lymphocytes.
Yes. It is one of the most stubborn, difficult-to-treat models available. It perfectly mimics human TNBC.
So what happened in the trial?
Well, when they administered just the anti-PD-1 therapy, the results were dismal. The Treg shield held.
The soldiers still couldn't get on the battlefield.
Right. Then, when they administered just the anti-TSPan antibody, they saw a partial response. The shield weakened, but the tumor persisted.
But the combination?
The combination was profoundly synergistic. The anti-TSPan8 antibody shattered the Treg shield, completely dissolving the localized safe zone.
Which allowed a massive influx of CD8 killer T cells back into the tumor microenvironment.
Exactly. And once the killer cells breached the perimeter, the anti-PD-1 therapy activated them to their full cytotoxic potential.
The ultimate 1-2 punch. Break the shield, arm the troops.
The combination drastically reduced tumor volume, halted metastasis, and significantly prolonged overall survival in the models.
That is incredible. But, you know, as compelling as mouse models are, mice are not humans. I mean, we've cured cancer in mice 1,000 times over.
Very true.
How did the researchers bridge the gap from a 4T1 model to actual human clinical relevance?
To validate this in humans, they conducted an extensive tissue microarray analysis.
A microarray. That's where you take tiny core biopsies from hundreds of different patient tumors and place them all onto a single slide, right?
Yes. It allows researchers to test the prevalence of a biomarker across a massive population simultaneously. And in this case, they evaluated samples from 730 TNBC patients.
730. That is a highly statistically significant cohort.
Very.
So what did the array show?
It revealed that roughly 22% of human TNBC tumors are naturally highly enriched with this specific TSPAN8 positive cancer stem cell subtype.
Wait, really? So nearly a quarter of all TNBC patients have tumors that rely explicitly on this non-canonical topological brainwashing mechanism to survive?
Yes. And when they cross-referenced that biomarker data with the patient's actual clinical outcomes, the reality was stark.
What did they find?
Patients who had elevated TSPAN8 and CD44 levels, which indicates a dense presence of these stem cells, they exhibited a significantly lower pathological complete response rate to standard therapies.
A lower PCR rate. Let's quantify that. What is the actual survival gap there?
Okay, so patients without the enrichment had a PCR rate of about 51%.
Okay, about half.
But patients with the TSPAN enrichment, they had a response rate of just 14%.
14%. That is a staggering drop-off.
It really is.
But, looking at it through the lens of this study, it makes perfect sense. I mean, we now understand that we've been trying to treat that 22% cohort with standard weapons, completely blind to the fact that their tumors are surrounded by a self-replicating, invisible shield of hijacked peacekeepers.
Exactly. The presence of TSPAN8 isn't just an interesting biological quirk, it is a vital, measurable biomarker.
It predicts, with high accuracy, exactly who will resist standard immunotherapy.
Yes, it embodies the true promise of precision oncology. You identify the precise microscopic mechanism of resistance, this secret handshake, so to speak, and you design a molecular wedge specifically tailored to break it apart.
So let's do our final quick recap for the clinical side of things.
Let's do it.
Number 1, anti-TSPAN8 antibodies act as superglue, blocking the cancer's EVs from talking.
Yeah.
Number 2, combining this with anti-PD-1 therapy creates a highly effective synergistic attack. Break the shield, arm the troops.
Right.
And number 3, this is highly relevant right now for the roughly 22% of TNBC patients whose tumors rely heavily on this exact shield.
A perfect wrap-up.
When you step back and look at the entirety of this proteomics map, from the weed-like roots of the stem cells to the floating skeleton keys bypassing the endocytic pathway, right down to the physical bending of the LKB1 protein that triggers the echo chamber, it completely shifts your perspective on what a tumor actually is.
It really does.
You have this tendency to view cancer as this brute force entity. We think of it as just wildly mutating cells blindly smashing their way through surrounding tissue.
Right, just a chaotic mass of uncontrolled replication.
Exactly. But this research proves that view is fundamentally inadequate. The tumor isn't just growing, it is negotiating. It is actively and aggressively manipulating its ecosystem. And it's doing so with an evolutionary nuance that borders on, artistic.
It really is elegant in a dark way.
It learned how to reprogram the defense forces without even needing to inject its own mutated material into them. It relies purely on the physics of surface topology. A glancing touch on the outside of a membrane is enough to reach all the way down into a cell's nucleus and completely rewrite its identity.
Which leaves us with a necessary and perhaps slightly unnerving final thought to explore.
Oh, I'm ready. Lay it on us.
Well, if an aggressive tumor can evolve to utilize non-canonical juxtacrine signaling, a completely secret topological handshake to perfectly reprogram our immune cells, we have to ask ourselves, where else is this happening?
Oh, wow.
What other systemic diseases, what mysterious autoimmune disorders, or even what vital normal biological processes are relying on this exact same invisible communication protocol?
Right, because we've spent decades assuming the only way extracellular vesicles deliver a message is by being swallowed.
Exactly. Now we know a simple touch is enough. It makes you wonder how many other microscopic skeleton keys are floating right under our microscopes, just waiting for us to recognize the locks.
That is a fascinating and, a slightly terrifying thought. It fundamentally changes the way we look at cellular communication. Well, we're definitely going to keep tracking the clinical trials on these anti-TSPAN8 antibodies as they move forward. A huge thank you to everyone listening for bringing your curiosity, engaging with the complex mechanics, and taking this deep dive into the science with us. Until next time, keep questioning the consensus.